right, biologists, let's talk about nucleic acids. This is theme A, unit and diversity, topic 8.1 molecules. So DNA as the genetic material of all living organisms. DNA stands for deoxyribonucleic acid. It's this very long molecule that we all have, all living organisms have. It encodes the information that makes the organism, right? So it is shared by all living things, and that points to a common ancestor. So we have here the DNA appearing first, and then this being shared with all the ancestors, all the organisms that came from this ancestor that had DNA, and now we all have DNA because of that. It's like heritage. Viruses, you may say, what about viruses? Some viruses have DNA, some viruses have RNA, but for the purpose of our course here, viruses are not organisms, they're not alive, so that's okay, right? DNA as a, whoa, let's go back. DNA looks like this, a double helix. It has, we call it double helix because it's helicoidal, but you have two strands, and then you have the bases in the center. Each of the steps here of the DNA, let's take a look again. Each of those steps, let's choose orange here. Oh, there we go. This section here of the drawing, this is what we're going to call a nucleotide. DNA is made of many of those nucleotides on either side of the strands, either side of the double helix. So this is what a nucleotide looks like. Do you have to memorize how to draw this? No, you do not. What you got to do is you got to identify the parts, right? So this right here. Oh my God, what's going on today? This right here is a phosphate. Okay, so you have a phosphate. This down here is a sugar with 5 carbons. Okay, so this is carbon number 1, 2, 3, 4, and 5 up here. So this, we can represent like this. This is a pentose. Penta meaning 5 in Greek. Pentose has 5 carbons. Now, this pentose may be a ribose or a deoxyribose. In this case, this is a ribose because of this OH here. DNA does not have this OH, but that's enough for now. And the rest here of the molecule, this is the base. Okay, this is the base. This whole thing here is the base. DNA has 4 bases. RNA has 4 bases. And you do have to know how to draw a diagram of this, not the exact molecule, but a diagram. So your diagram is going to go phosphate. Here are pentagon, our pentose, and then the base, right? Know how to draw this. This is important, right? And then you have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, forming what they call the phosphate deoxyribose or the phosphate sugar backbone, right? It's like the outer part of the strands, and the rungs of the ladder are here, the bases. You have adenine, thymine, guanine, cytosine, and DNA. So the sugar phosphate bonding and the sugar phosphate backbone of DNA and RNA. So both of them have this, but when you have one nucleotide, let's draw our nucleotides here. You have, my God, you have our phosphate, we have the pentose, and we have a base. Then you have another nucleotide. Let's make a different base now. Let's make a purple base, lilac base. There you go. How do you connect these two? Well, with a covalent bond. I'm gonna make it green. The covalent bond here between the two nucleotides. It comes here from this carbon to this phosphate, right? It's not some random connection. It's gotta be like that. So when you add a new phosphate, where are you adding the new phosphate? To this one. Okay. So this will form right here the sugar phosphate backbone. And the bases in each nucleic acid that form a basis of a code. So DNA has guanine, cytosine, adenine, thymine. You can remember by ghosts. Ghost cats are terrifying. And RNA, a little bit different. Ghost cats are ugly. Ghost cats don't really like this, but you got to remember, right? So guanine, cytosine, adenine, and uracil looks a whole lot like thymine, except it doesn't have this methyl group down here. And now we know that guanine pairs with cytosine. We know that adenine pairs with thymine. And now you can see why. You see the dotted lines, whenever you have to represent them, represent as dotted line. These are hydrogen bonds. We learned about hydrogen bonds before, right? So we know that guanine pairs with cytosine because the hydrogen bonds here fit. If you try to put a thymine here, the distances between the atoms will not work. The whole thing will not be stable. Oops. RNA is a polymer, right? Formed by condensations of nucleotides. To be fair, DNA is the same thing. DNA and RNA both are polymers formed by condensation of nucleotide monomers. The point of the slide here is to remind you what condensation is, what a polymer and monomer are. So RNA, similar to DNA, is ribonucleic acid. It's also made of nucleotides, but then we call them here monomers because monomer is the smallest brick part, like the smallest, smallest Lego brick. It's the smallest part that you add many of those together, you make a larger molecule, right? So polymer, when you have like polyethylene, well, you have many ethylenes connected together by covalent bonds, making this larger molecule. Ethylene itself is one molecule, but polyethylene is like this super structure made of many ethylenes all connected to each other. Same here goes with RNA and DNA. You have one nucleotide, but then when you connect many of them, when you saw how they're connected, the sugar phosphate backbone, now you have a polymer, you have a larger molecule, you have a larger RNA or DNA molecule. Now, condensation is the reaction. We call it an anabolic reaction. Every reaction that builds something is anabolic. If it breaks in biology, if it breaks, it's catabolic, right? So it's an anabolic reaction because it's going to take a smaller nucleotide molecules and make a larger, and make a larger DNA or RNA molecule. So what happens is, the OH groups here, they will look at each other and say, hey, you have two hydrogens and one oxygen. This looks like a water molecule. And this water molecule will be formed as a subproduct. But now you need to finish here this covalent bond because you remove the OH, and here the oxygen is missing an H as well. So neither the oxygen or the carbon is happy. So you made here the covalent bond. Now everyone is happy, except me because of the string. And then you have the sugar phosphate backbone being formed, removing a water molecule. This type of reaction is called condensation, and it happens all the time between carbohydrates, between amino acids. This is usually how you go from monomers to polymers in biology. Oh my, DNA has a double helix made of two antiparallel strands of nucleotides with two strands linked by hydrogen bonding between complementary base pairs. Now we know already what complementary base pair means. It means that thymine goes with adenine, right? Thymine pairs with adenine. Cytosine pairs with guanine. And we know why, because of the hydrogen bonding between these complementary pairs. One always goes with the other because that's how the hydrogen bonding fits best, right? It would not work if you try to pair thymine with cytosine. The distances between the atoms would not fit. The two base pairs, they would just not fit. It would be super awkward. Now, the antiparallel part of this thing is what can be a little bit confusing. Now, see how the oxygen, gosh, the oxygen is up here. I'm gonna use a different color. There you go. Oxygen is here, oxygen is here. This carbon is pointing up, this carbon is pointing up, and you always have this carbon and this oxygen pointing up, linking to the phosphate down here, etc., etc., etc. Now here they have a different situation. The oxygen is pointing down, the carbon is pointing down. Oxygen is pointing down, carbon is pointing down. If you look, I have phosphate, sugar with the oxygen pointing up, phosphate, sugar with the oxygen pointing up, running this way. Here, you would have your phosphate up here connected to the carbon, and then you have the sugar with the oxygen pointing down, and you have the phosphate, and you have the sugar, and you have the phosphate, etc., running the opposite direction. They are parallel, they are running side by side, but one is upside down and the other is right side up in relation to each other. That's what antiparallel means. It means they are parallel. They are running side by side, but one is upside down in relation to the other. Now, the role of complementary base pairing in allowing genetic information to be replicated and expressed. Replication is when you have a molecule of DNA and you make another. And the way this is done is very elegant. The experiment that showed that this is the case is also very elegant. What happens is, since the bases are complementary, A always goes with T, you can just separate the double strand into two single strands. And well, if I know that I have an A here in the single strand, I know that on the opposite strand, I must put a T. If I know that I have a C here, I know that the other one has to be a G. So one strand serves as the template for the new strand. The old strand serves as a template so you can build a new strand. So this is a very economical and elegant way of replicating the DNA. You don't need to know how to make a DNA like from scratch. How do the enzymes know what base to put next? You have so many bases. Well, you just have to pair with whatever already exists. So that's brilliant. Gene expression, on the other hand, is usually taking that gene, that segment of DNA, and making the final product, which usually is a protein, right? So you have the DNA, you have a process called transcription that makes our messenger RNA, and from the messenger RNA, you have a process called translation that gives you the final protein. In some cases, the final product is an RNA, like transport RNAs, tRNAs, for example. They are the final product. There is no protein that they encode. But most of the time, the final product is going to be a protein. And genetic information, is just, parts of the DNA that encode the instruction. In eukaryotes like you and I, not all of the DNA will encode instructions to how to make proteins or RNA. Some of it is there for reasons that we are going to explore in other subtopics. Now, the DNA allows for an enormous amount of combinations, right? It's ridiculously astronomical, the number of combinations that you can have with just 4 bases. So if you only have 1 basis, you have 4 possible combinations, right? You can either have A or T or C or G. But if you have 2 bases, you can have these 4 times the other 4 of the other base. So you have 16 possible combinations. If you have 5 bases, it's 1,000. You have 10 bases, more than 1,000,000. 15 is more than 1,000,000,000. And tRNA, the smallest gene that we have, right, that is only 76 bases long, you can combine 76 bases in more than 100 trillion times the estimated number of stars, in the entire universe. It's insane. And yet the entire genome of a human fits in a CD. In those CDs that we used to have early 2000s, that's it. That's like about 800 megabytes, the entire genome of a person. And yet you can store so much information there, like limitless combinations for what we can do. And that is absolutely genius. And it all fits inside the nucleus, inside a tiny, tiny cell. All right, people, this is it for today. This is the standard level content. We are going to have a separate video for the additional higher level stuff.